Each of you mentioned that purification is an area ripe for more discussion. So I'd like to sort of take the pulse check of the world of purification here. What is the single biggest challenge you're facing, right, in the purification of your mRNA, let's say drug substance? We'll focus on that for now, drug substance today. And maybe we can start with Mark and move to Christian and then Stu.
Yeah, I would agree with what I think Christian was the one that really said it at the beginning, is there with chromatographic purification methods, you're often limited to the scale because of the column size. And so you're, depending on what scale you're trying to tackle, you're limited on the scale end, and then you have to start scaling out and running repeated unit operation cycles. So, I mean, there's ways to address that. You know, the pure upstream process or product that you bring into that, that would help. So sometimes it's a holistic approach to process development. It's just not focusing on one unit operation, in this case purification, but also unit operations that are feeding into that and impact that.
Yeah. So focus on the upstream, right? All of the other aspects that come before it to try and get to the purest drug substance that you can to help negate that.
Maybe because I think you really have to look at it holistically. I don't want to jump topics here or anything.
Please, go for it. You look ready to say something. I'm ready for it.
The logical step in process development, right, is, hey, the purer we can make the material upstream, the easier, the less pressure we're putting on the downstream purification processes.
Yes.
I think we're in a unique situation in that we're still, the vaccines have been incredibly successful. However, the RNA space, I still don't think is well established in the therapeutic space. We're still a new entity here.
Absolutely.
And so what that usually associated with is really high cost reagents and raw materials. And so there's a cost to getting pure material upstream, and it might be a cost in yield, it might be a cost in lost materials through the process. So I think you have to balance that all out. How much is it costing me, and what type of yield am I getting on the upstream side if I completely optimize to purity? Versus, well, what if I don't optimize to purity, but I hit some type of sweet spot with purity because it's actually cheaper for me to invest more on the downstream purification process in terms of cost. So it gets kind of complicated. That's why I think you really have to look at your whole process holistically, and the performances of each of your unit operations and the costs for those unit operations, and then match that out. What's the best combination of those to drive the final yields and the final costs?
Interesting. See, I like this. I find this an interesting argument, right? Because again, it does kind of go against that normal ingrained start out, put all of your investment in your upstream so that you lighten your downstream load. But in this case, it seems like the trade-off could also be maybe it's better or more affordable to focus on the downstream to some extent in this case because of all of the innovation that's happening with reagents and raw materials and whatnot. Christian, do you agree sort of with Mark, or are there other comments that you'd have in terms of some of the challenges that you're seeing with downstream purification in particular?
Yeah, I would absolutely agree with Mark. Of course, it's great to have a high purity product after upstream processing, but if you have a robust downstream process, that definitely is worth investing in. As mentioned already in the beginning, it's a burden with scalability, and if you're aiming for larger patient populations, and here at Ethos we are developing a nasal application in the field of asthma. So it's really large patient populations where we're aiming for. We need large batches, and we knew that from the get-go. So we invested in upstream as well as in downstream, and so took a quite close look into that. And we at the end could come up with a very efficient purification strategy that comes along without any chromatographic step. We just purely work TFF based. It's several TFF steps, not just a single one like you do with diafiltration or concentration step, but it's involving certain buffer systems, and that has shown us excellent performance. We get really pure RNA out of that. Worked nicely with us. We did that internally, developed the process, could also transfer it to CDMOs and produce GMP batches on that. Have discussed that with regulators as well, and they are absolutely fine with the quality that's produced there. And it's not a trade-off, but it's more a development perspective. And yeah, we have a very good CDMO with that. I'm not making any special comments, but with Thermo Fisher, we have it there, and we think it's worth providing that technology also to third parties, and that's possible via our CDMO. So happy to get people in contact on that topic.
Okay. Awesome. Stu, what do you have to add here? Is this something that you also are struggling with? I mean, Mark really nicely outlined some of the biggest challenges we have, right, with downstream purification. And so what are some of the biggest challenges that you're encountering? And are there ways that you are also, and Christian was talking about some of the ways that they've optimized, right? They're streamlined their downstream process. What are some of the, sort of novel purification technologies maybe you've been able to play with, or some of the novel purification process flows you've been able to check out, right? That are, you think, hold some promise for the field at large?
Novel purification.
Anything exciting out there?
There's not too many that I'm aware of right now for purification. I know there's a lot of dsRNA removal type resins that are hitting the market, especially like flow through type columns where you don't it's not a bind and elute type chromatography, so that kind of makes it a little more scalable and less complicated. So things like that. One of the biggest challenges too we get hit with is like removing truncated RNA. So especially with larger RNAs, you're going to get a lot of hydrolysis products, aborted transcripts, things like that. So your RNA integrity won't necessarily be as good as shorter RNAs can, because short RNA, you can come straight out of the IVT, maybe you've got 90+% is the full length product, whereas with larger 6 kb plus mRNAs, especially like self-amplifying RNAs, get really large. You can come out of the IVT with much lower purity, and so removing those truncations during downstream is really difficult. So I guess that's one of the biggest challenges we're facing too there, is removing those truncations, because they're hard to separate, right? And it's kind of the same material as your full-length products, so there's not, it's really difficult to get that out.
To separate those out.
Yeah.
Interesting. Are there, this is perhaps a question for the full panel too. So Christian, you just outlined that you've managed to eliminate chromatography, right, from your downstream purification process. But, I'm curious, what other opportunities exist for process simplification, in terms of eliminating or maybe consolidating steps, particularly in DSP, to help improve efficiency and reduce our cost of goods? Are there other methods, right, that you guys are watching in this space? Maybe Christian, we can start with you and go to Mark and then Stu.
Yeah, that's a good question. So I think very, very new and very early work is been doing on continuous manufacturing, on a lot of inline analytics and online analytics, where you really during batch manufacture do constant analysis of the conditions, and you get a lot more control on your process. That might help in reducing impurity generation and more efficient use of the material. So if you do not have too much excess of enzymes, NTPs in there, you might be able to also simplify your downstream processes at the end. But I think much of it is very early work, and this will take some time until this translates into products or in product candidates. I think the classical strategy with the upstream bioreactor and the downstream with TFF and potentially a few columns in there is the best you can do in the moment.
Yeah. Stu, what do you think? You're nodding.
I kind of agree.
Dangerous. I know it's dangerous. She's like, she sees me nodding and thinks I have something to say.
Yeah. If you want to drive the yield up, oftentimes you're adding a lot of raw materials into your IVT too, and so that gets expensive, especially if you're using licensed materials, cap analogs, things like, especially the cap analogs and the enzymes can get really expensive. I know I suggested engineered polymerases, but they're incredibly expensive, making up the bulk of the cost of the raw materials. So I guess you just kind of have to optimize and see what you can get away with, on yield and purity in your CQAs and go from there. But yeah.
Mark, what do you think in terms of some of the, are there opportunities that exist for process simplification and downstream that you're watching as it relates to the world of circular RNA?
Yeah, I think from a downstream perspective, it's really understanding what's an acceptable purity profile.
How pure is pure enough, right?
Yeah, because the more pure you make it, the bigger hit you're taking on yields. It's very difficult to separate or to purify, I think, RNA from RNA related impurities with standard approaches. And so often you're, maybe pulling fractions, and so you just have to be careful. You don't want to make it too pure where it doesn't have an actual impact on what you need the product to do, and coming at the cost of throwing away a portion of your drug substance in that purification process. Okay. So, yeah, and I also agree with Stu. I think one of the ways right now, I think you had a talk or something with someone recently where the cost of materials could be estimated up to 75% for RNA products.
Yeah, exorbitant.
It's worth the while to really take a look at your bill of materials, find out what you're spending the most on and how much is going into your process, and then going back into your process and maybe optimizing around yields and conditions around the use of those really high cost materials.